So I'm going to share my screen here. Let me hit OK, not leave meeting very carefully. Um, Fantastic, thank you. I'm going to share my screen here, and let's make sure I share the right screen. There we go. I've got just a few slides here to talk about what how to grow almost anything is, and then I want to talk a little bit about how we're structuring the use of the Opentrons, both for our collaborative project as well as for the labs within the course. And my dream goal out of this discussion is that we find ways of making this better, and we find ways of making it applicable to a broader audience than just the course, and that we've got an opportunity here with 10 to 15 global labs having an Opentrons and an interest in bio activities throughout the year, not just around how to grow almost anything, to really use this to seed an open lab automation effort in a way that we haven't been able to before the kind of donation that Opentrons is making available for this course. So a little bit of structure on the course. This is run by David Kong, George Church, and Joe Jacobson at MIT and Harvard, and has about 20 to 25 MIT Harvard students every year, and way more than that in global students. And that's one of the things I really appreciate about the way that David has run the course, is making it open to a global listener community, either as a committed listener where you commit to doing all the homeworks and reviewing them with the TAs, or as just a general listener who doesn't really commit to doing anything except for watching the lectures. So we had 500 remote participants at the start of this year, and it's dropped off a little bit now, but it's still in the 150, 200 kind of range at a lot of the lectures, and so it's a big thriving community that follows along with how to grow almost anything. Been going on since I don't know, Scott, you were in one of the first cohorts. I think that was 2016 or so, wasn't it?
2016, yeah, it wasn't the first. It was, I think, the third. Like the first two just in-house. So I think ours.
Yeah, so been going on a while and getting bigger all the time. It was loosely modeled over how to make almost anything, but very, very loosely. Yeah, the detailed description of the course is in a Nature Biotech article from a couple of years ago, and is really around this idea of lectures supporting labs, supporting projects, and so the final individual project is one of the key focuses of the course, but this year is being supplemented by a group project, a group collaborative project that I'll talk about in more detail in just a minute. You guys are all familiar with this. Was just pulled from a different slide deck, but want to set the context of where these robots are going. They're all going in community labs, and in many cases, community labs associated with the university lab, but not on the university campus. And so we're in makerspaces and hackerspaces and GenSpace and BioCurious, my tiny little robot corner at Makerspace from last year. That's Scott's Opentrons. We're getting one of the new allotment this year. So we're really kind of chaotic community lab spaces that we all know and love. The effort's really a very global effort, and so here's our map of this year's participants. Can see popping around from all over the world. Everywhere you see a robot poked up here, that's a site that either has or wants to have a lab robot. So see, we have pretty good distribution. I think we're missing robots in a number of the US Canadian sites, but pretty good distribution over these global labs that will have some automation capability. The main thing, I guess, that I want to focus on in here is final project and how that's set up and how that's driving some of the automation requirements for this network of robots, because one of the requirements for people in this year's robot distribution, and Opentrons has indicated a willingness to consider future distributions as well. They really want this to grow. It's going to be 10 or 15 robots this year, and if we show we can use them well and effectively, they'll continue seeding that in future years. And so one of the requirements for the folks in the course that put in an application for a robot this year is that they support the final project, which is being led by a postdoc in George Church's lab on bacteriophage engineering. And taking the MS2 phage and engineering the lysis protein. One of the things that's very interesting about MS2 is it's a very, very small genome, 4 genes, about 3,600 base pairs. And we're taking one of those genes, the lysis gene, and attempting to engineer away the dependency on a chaperone protein within E. coli, so as to make it more applicable for some display phage applications and other places where resistances can occur through the requirement of that chaperone. So what we want to do with the global network of opintrons is how do you fertilize opintrons? I guess opintrons is to construct a fitness landscape for the mutations that we'll be using. Twist has given us about a 20K synthesis budget. So with it being such a small genome, we've constructed some plasmid designs that allow us to produce quite a good number of mutants of that license protein. And we want to have all of the labs with some automation be evaluating these mutants with the goal really of ensuring that we can have a consistent application of a protocol across remote sites on the opintrons. We're going to do some initial screening on the Nuclera platform, which is an interesting platform for protein engineering. This is using a microfluidics chip and looking at the agglomeration of the proteins as a way of telling if they've been properly folded, so we can discard some of our mutants right off the bat if they don't fold properly and not have to run them through the entire set of assays we do remotely. We are going to use plaque assays as our primary measure of the infectivity of the bacteriophage, but we also have a bit of work going on to try and do some metabolic monitoring. And this is where I think it's really interesting to try and extend the sets of tools that we have available on the opintrons. You can use a glucose meter for diabetics for saying the glucose content in liquid culture with a little bit of calibration and mucking with it. But the off-the-shelf tool for measuring the oxygen content, which is the other element of metabolic monitoring, is a couple $1,000. And so we found some DIY plans for using a redox sensor and gold wire to make a home-built oxygen sensor. And so we want to try and rig something up to allow us to do metabolic monitoring and liquid culture as compared to having to plate and do plaque assays for evaluating all this. In reality, we'll do both and may or may not get to the full metabolic assay this year. But I'd love at some point to bat around some ideas on metabolic monitoring on a lab automation platform that is not a bioreactor. Plainly, this is something that bioreactors do really well, but something that cultures that we're just doing in a growth chamber on a shaker plate inside the opintrons do much less well. So that's probably where I ought to jump into what equipment we actually expect to be at each of these sites. We're going to get the OT2 itself, a P20 and a P300, and then temperature module for doing the cooling cycles, being able to hold reagents and enzymes and stuff on the bed without needing to constantly shuffle them back to the freezer. Thermocycler module, so we can do a PCR and related protocols on the OT2. Magnetic module. This is for allowing us to use magnetic bead protocols for separations. And then finally, a heater shaker module, which we'll be using for the actual liquid culture growth on the OT2. Plainly, the modules that come with the OT2 are, as soon as you get any number of them, way more expensive than the OT2 itself, and with much less justification in some sense. And so one of the things that we're going to want to do as we extend this is use some of the manufacturing capabilities that we have in our network of hackerspaces. We've got a surface mount technology pick and place machine at Victoria Makerspace, and some other spaces have similar types of manufacturing capability to build our own modules that plug into this. That's one of the places that we probably, as we get into the discussion portion of this, want to talk to Rick about this trade-off on using PyLab Robot versus the OpenTron's native protocols, and how we actually extend the automation software to handle new modules that may not exist today. And so that's really kind of the extent of the prepared discussion that I had, or a prepared presentation I had on what it is that we're doing with How to Grow Almost Anything. And I wanted us to hit a whole bunch of questions around this, including how we bring people up to speed and train them to use the OpenTrons. And I should have put this into the slide deck, but didn't think about it until we were talking just a moment ago. But the way we've done it this year is with a couple of labs described in the Google Doc I've just thrown in chat that go through some colored plates and moving colored E. coli around and doing a fluorescent bacteria and black agar lab on top of the OpenTrons. So yeah, let me kind of leave it there and open things up to this roundtable discussion format. And maybe Tim, I could get you to provide a little bit of structure in how you think we ought to handle that. Shall I ask some questions or start some discussion? Where do we want to go?
Yeah, no, that sounds great. I mean, I think maybe we just kind of, let's throw out the first question and see who jumps in. I think with this crowd of people, there's some experience getting labs set up and dealing with this. So let's see how it goes. If you want to just, you can sort of facilitate.
Sure, sounds good. And Scott, you're right. We talked about the HEPA unit for the OpenTrons, and it was one of the ones we pulled off the list because we were trying to get OpenTrons. They'd give us an initial budget of $500,000, and we decided, oh, we can increase the number of modules and reduce the sites, or try and get more sites and reduce the modules, and for a HEPA filter and a fan to plop on top and not interact with anything, it seemed a crazy price to pay. So I'm curious how your experience has been on printing and testing. Are you getting good sterile conditions?
Not yet. So I'm just waiting to get the 3D printer in, and I'll print the holders for the fans, the computer fans, and I've got Lordco just across the street to pick up different types of HEPA filters to see what's best. But that's a low-cost way of doing it. A friend of mine works at a company that makes a commercial 3D printers, and they're doing exactly the same thing. The 140 millimeter diameter fans, computer fans, hooked up to automobile filters. That those HEPA filters are really cheap. So yeah,
it makes it shocking that their module is so expensive.
Yeah.
Cool. So there's the physical. I'm sorry.
Sorry, Derek. For the first question that you have there, I was wondering, how did you have it set up last year when you had my OT2?
Yeah, so there are a couple of aspects of how do we get remote labs set up. The first is just physically, how do we get the machines there, get them installed properly, get the shipment figured out, get them with supporting equipment all figured out. And so when we did the application for machines, we asked what other equipment was going along with the machines. So if people had a -80, if they had a microfuge, if they had kind of all the other little bits and pieces that you need to do for off the machine bits of the protocol to make it really usable. And when we did our robot site selection, we prioritized those locations that did have supporting equipment and did have a reasonable number of How to Grow Almost Anything students. We're still having some problems with getting the actual distribution of machines out to the different locations. All of the US machines are going to ship from the OpenTron's US warehouse, and so there's no real problem there. But the international ones are going to ship directly from China, and thus, of course, incur import fees that in the case of Canada, anyways, we're coming up to about $7,000 for per machine, simply because that full retail price of the machine and all the modules, which they can't really legally waive on much, gets pretty expensive. Our workaround that we've found for this is that in most of our sites, there are a couple we're still trying to find solutions for, but most of our sites have an associated university, and so we're getting a donation letter. Not clear if it's coming from MIT, which is where OpenTrons wants to directly donate the machines to, or if it's coming from OpenTrons itself, to each of the associated universities that can receive the machine and then forward them along to the community lab. And so in the case of the University of Victoria, we're going to really just kind of skip through purchasing and procurement and everything at UVic and have me just fill out the shipment receipt form, and with that donation letter, which will say that this equipment is donated to the University of Victoria for placement in community labs for support of the How to Grow Almost Anything course. That will allow us to basically just hand it off from the university to the community lab. And with the university being a charitable institution, universities don't pay import tax, and so that will get it handled there. We have a similar setup for sites like the one in Ecuador and Thailand and Japan, but some of the other sites don't have an associated university, and so we're still trying to figure out how to get it to work such we can get the machine to them without incurring huge fees.
For do the other sites have all that supporting equipment? You know, the -80 is sort of the first one to come to mind, but it's also a laminar flow hood is really helpful. An autoclave, like all this stuff. Have you been finding that those things exist? Because to expound just for a second, if we're talking about remote labs distributed, I think sort of built into that concept is the idea that it's not that smaller labs can work, and so they need to be cheaper labs. And like, you know, the OpenTron sort of points towards this exciting idea that actually for a few $1,000s you could, you know, start to do something. But then when you add all that other stuff in, it's still, you know, $50,000, $100,000, which is a lot of money in a lot of places. Have you found that to be true? Or?
No, that's right. We have when we did the application, we asked specifically for -80. Here, let me pull some of our responses back up here. Probably not. Yeah, so here is the list of things that people have been replying to, and these were checkboxes on the application form. Is how we got these, and so we asked for the -80, -20 fridge, a tube rack scale, purified water, electrophoresis, microfuge, vortexer, incubator, gel imager, spectrophotometer, microscope. PCR equipment, and I think there were a couple things on other things on that list. I don't remember exactly what. We asked about laser cutter and 3D printer on that list because we're going to 3D print some of the appliances that go into that. And we then prioritized the sites that we're going to hit in this first round by those who had the appropriate equipment to support the robot. I think one of the goals as we talk about how do we actually make this distributable in subsequent rounds to sites that may not have all the supporting equipment is just how we build supporting equipment to go along with this and how we bring those costs down. We're not going to build a -80, of course. That's not really a DIY-able component, but the small chest freezer, not big ones, but the ones that are like the coolers you put in the back of your car. You can get a -80 version from Alibaba for about $2,000 now, which is sure a lot cheaper than going through ordinary lab procurement mechanisms for a full-sized one. So I think that if we can use some community intelligence to try and figure out what the lowest cost way is to fully support a future year robot distributions, we can bring the ancillary equipment requirement down over time.
I have a question for Nadia, kind of on that note, because I know, Nadia, that you're not fully specialized in the synthetic biology world. You do kind of a lot of different automation and human-computer interactions. Like, is this pain point, from just like, the experience that you've had, is this pain point much more prevalent in the bio world? Or, because like a 3D printer, well, you can buy a 3D printer, but I don't know. I mean, I don't know if that strikes you at all.
I think a lot about the differences of setting up different kinds of automation things. And I think one of the things that, like, from the outside, you're like, oh, they're machines and they do things, and engineering is the same, and you buy them and you customize them, and it's no big deal. I think one of the huge things that's really different and makes a lot of the distributed and remote stuff hard with bio is that you're dealing with living organisms who have this pesky tendency to die. And so it's like, when you're collaborating with other people, if I'm collaborating with someone else on my 3D printer and I'm like, oh, let me coordinate this with you, I'll do this, and then you do this some amount of time later, whether or not that happens now or that happens next week is kind of not a big deal. Whereas if I'm like, hey, can you change out the media? If you don't do it on time, everything dies. And so I feel like this remote collaboration and distributed outfitting, it sort of really prioritizes something very different depending on the application space. And so when you're like, oh, can we do a cloud lab or remote stuff? Sometimes I feel like centralization becomes more important for bio applications because you need to have this tight control of timing. And the other thing that I think is, like, I think in digital fabrication, 3D printing filament is super cheap in comparison to the machine. But for bio reagents, I don't know, I bought this custom DNA. It's much more expensive than the machine is actually working on, doing something on. And so the risk profile is really different. If you're like, oh, I'm going to mess up this sample, if you're 3D printing something, you're like, whatever, I'll just print it again. Whereas if you're messing up a sample where you're like, oh, I've been working on these for years or months, or I don't know, for a long time, then the trade-offs are really different. And so, I don't know, when designing systems and designing machines, it's something I think about a lot, and it's sort of hard because at a high level, you're like, oh, it all looks the same. It's like a XYZ Opentrons. It's basically a 3D printer. But the things that people do, I don't know, it's really, really different, and therefore how you design for it and the risk. I don't know. You guys all know much more about this than me. I don't know why I'm talking so much.
No, that's totally good points in there. I think one of the things we're trying to do with this is, and David and I just worked on a JWell grant to try and get about 100K to support reagents and materials to ship to these remote sites. But trying to make the investment in the materials around it for the group project low enough individual expense to let people fail a few times while getting protocols to work. And that's really hard. If you're going to do a 10K synthesis at Twist yourself, you don't have any chances for mistakes. Twist is actually willing to split our syntheses so that they continue to grow up a larger quantity after they produce it and then ship it out for us to the other labs, which got us around a huge problem of we can't ship any genetic material from MIT or Harvard because of material transfer agreements. But if Twist will do the splitting up for us, we're under a Twist MTA, not an MIT MTA. Yeah. And so getting the package of consumables and materials to support the experiment to each of these labs, so that they can practice is one of the key things that in terms of lab setup, I wanted to get thoughts on. Maybe we should talk a little bit about that kind of training and getting people up to be able to do protocols together. And Rick, I would love to get you to talk a little bit about PyLab Robot and how that sits in relation to the OpenTrons API and just kind of what we, as not just a how to grow almost anything community, but really a larger lab automation community that I hope this spurs, how we figure out how to do a shared protocol library that might be extensible by different sites.
Sure. So I guess I'll very briefly give a general description of PyLab Robot and then talk about how you can use it for OpenTrons. So PyLab Robot is this open source hardware and OS agnostic interface to lab automation devices. So we have universal interfaces for example, liquid handlers and also other machines like pumps and skills, where you can use the same API to say like aspirate, excuse me, dispense, pick up tips, etc. And then there are different backends that are going to translate this to commands that specific robots understand, such as Hamilton robots, TCANs, and also OpenTrons. Now, OpenTrons already has this Python API, so there's less of a value add in this case. But for Hamiltons and TCANs, for example, they normally come with this very complicated GUI program, and so PyLab Robot really adds a ton of value there. I would still say for OpenTrons, there is some value to be added. The one major thing is that for OpenTrons, when you run a protocol, you actually upload your Python script to the OpenTrons onboard Raspberry Pi, and then the Raspberry Pi is going to execute your protocol for you. Which I guess is nice because you don't have to have your computer next to it, but this can also cause problems because it is less interactive. And so with PyLab Robot, actually, you just use your own computer, and the OpenTrons serves almost as like a peripheral device where you're just sending commands to it continuously. And this is done using their HTTP API. And you could still, of course, use another computer to send these commands to the OpenTrons if you want to, for example, take your laptop away if you're using it for longer runs. And then second, an advantage that I would mention over just the normal OpenTrons software is that we actually provide this visualizer so that you can sort of do simulation runs without actually having to be next to your robot to see what is going on. And we found for how to grow almost anything here at MIT that it's very useful for students to sort of play with this around before they actually come into the lab and run a protocol on a robot, because they can write it and sort of play with it. And then the labs become much more productive in this way. I should mention that we support most of the features that OpenTrons does, but for example, the PCR machine, I've not integrated yet. We do have the temperature module, but yeah, if you wanted to use the thermocycler, I think you should let me know, and I can probably get that done within a day. But yeah, there are a couple of things like that where you probably need to request or write some code yourself before you can use them. There's a fair disclaimer.
Can I ask a question? In practice, for PyLab Robot, do you find that the reproduction of the script that's defined in PyLab Robot, you get similar results on, say, a Hamilton and an OpenTrons? Or, I mean, because they are physically different systems, which I think is one of the great challenges of distributed automation. But how does that work for you? Have you had issues?
So I would say that the protocols themselves, they just execute correctly. But I have to admit, I've not done like really real biology experiments that I've shared between robots. And so it's hard for me to give you like an answer from the biological perspective.
Cool. Thanks. One of the things I find really exciting about this is that we do have a fair amount of computing expertise in the distributed network, and so I hope we get some subset of that to be able to help in some of the module creation, particularly if we get to creating our own modules for this. Let me ask the question around other ways of ensuring consistency in a group collaborative experiment. So I think getting the robot to do reproducible timings, reproducible quantities is only one part of the battle. We've talked a little bit about setting up a webcam to let people remotely attached to the Twitch channel to see all 10 robots running their things at various points in time, and other cute ways of making it seem a bit more collaborative. But really, I'm looking for ideas on how to make it more reproducible. Automated monitoring, logging of conditions, maybe an electronic lab notebook we can all standardize around. And if we're running this in an automated way, we ought to be able to be a bit more reproducible. And if there is a problem in the biology, be able to go back and see what might have led to that problem, or if it is a problem in the biology as compared to the automation side. Anybody have thoughts or places that you would like to point me to on tools of that sort?
I guess one thing that I can say is that I found Jupyter notebooks extremely useful, and you should all be using them for as developing protocols, but also for running them. You can just set it to run, and if you're have a computer that's just sitting there, you can just leave it running in a Jupyter notebook. You don't necessarily have to write a Python file and execute that, because some people say that's the proper way. I think Jupyter notebooks are probably better. And you can also just include like prose, like Markdown. I found it a huge advantage.
You can change things as well. I mean, like we've done quite a lot of extraction protocols and mini preps and stuff, and like sometimes you can just tell it needs another wash step or something. Like you have a little bit of flexibility to just turn things around. Yeah, so we've had like, I don't know, we had a whole team of 16 MPhil students that were using the robot at some point, and so there's probably been about, they weren't all using the robots, but like, let's say at least 12 students have gone through and kind of learned to use it. And all of them have really enjoyed the Jupyter notebooks kind of mode of doing things, and just being able to kind of adjust things a little bit on the fly and try things out. So it's pretty much our default now whenever anyone wants to start using our OpenTrons, at least. It's more intuitive for people as well, especially the non-coders, of which, to be honest, most of the people that we've had try not really Python. I mean, they can sort of figure out what's going on, but they're certainly not. They don't know Python as a language, so it's a bit difficult if you haven't got the annotations and comments.
For remote monitoring and especially video capabilities, the easiest way would be to use the module from the OpenFlexure, which also has autofocus capability, and it runs on Python, and it uses just fairly cheap a Pi microprocessor and a Pi camera. I think it's 5 or 8 megapixels right now, and that would it's fairly light and fairly easy to place. The code has to be changed though. That's the only thing.
Cool, thanks. On the Jupyter notebooks, which I love the suggestion in general, I use notebooks a lot on in many things, but I'm really kind of wondering what the level of organization above that is. Meaning that if I have a well-developed protocol to take a twist mutant and run it through its series of steps to expand it, and then inoculate liquid culture from it, and then pull out of that liquid culture to plate it. And I've got 20 mutants for each of 10 sites to try it on. Now, just kind of keeping named Jupyter notebooks in a location to try and validate that here's exactly what happened, and a place to hold a note that on run 12 at site 5, you had this odd event happen. There's almost a level of organization of the notebook runs that needs to happen above the level of the individual notebook. And this is kind of what I had in my head around an electronic lab notebook structure, but I'm not aware of any tools that kind of fulfill that higher level need.
So it's like a lab notebook that you would put like a Jupyter notebook into, or something, like a larger sort of project organizer?
I mean, the way I do it as a computer scientist, of course, would be simply to check my Jupyter notebook into GitHub, and every time I run a run, I make a check-in push and add a comment to it as to what run that was. But this may not be as biology friendly. Introducing Git to non-computer scientists as your lab notebook is maybe a step too far.
I mean, I might just jump in here and say that given that How to Grow kind of has operated a lot with Notion and having people use personal pages, I find Notion to be very easy to use. And, you can attach different files, like including Jupyter notebooks. You can write code chunks, and kind of just feels pretty intuitive. So it might make sense to stick to that, but have like a shared, kind of protocol notebook or something, something in that system that might work well.
That's what I was going to suggest as well. But the downside with Notion is that it becomes very chaotic if everybody starts trying to organize in their own way. So definitely recommend setting up a structure for people to follow, because you can end up with 12 different databases that are trying to do something in different ways. But for consolidated structure where it's both very cheap or free, and you can host the files and add annotations to the experiments, you can create entities in a trackable way as well.
Maybe to add to this idea of using Jupyter notebooks, both as the sort of the recipe for what you run and for recording what happened. In my experience, I try to completely separate the input, like the recipe, from what you recorded as the output, because in any realistic lab operation, what happens does not, only matches the inputs in like 90% of cases. And those deviations, depending on what format you choose, Jupyter Notebook, for example, you might not be able to record them. So at least sort of in larger setups, I've always seen it as a failure mode and tried to just, different formats, different everything for inputs versus recording what actually happened for the log.
Yeah, I've sort of moved to a general philosophy of log everything. Like you just need to know every single possible thing that might have happened so you can figure out deviations. But the best way to do that I can think of right now is still Notion, where you just ask people to write notes, unless you want to set up a really robust system. But
yeah, I
Rick,
think maybe to phrase it more clearly, like the thing to avoid is that the only thing you're allowed to record about what happened is checking off lines in a recipe because something else might have happened. So sort of in terms of laboratory information management systems, like sometimes they are set up in this way where the only legal thing that you can possibly write down as what happened in reality is you followed the instructions, but what do you do if it's not the case? So if you want to record actual true data, you need to give more flexibility for recording what happened.
This is one place, and it's kind of silly, but one place that I was thinking of that camera. I mean, part of it was for the stunt Twitch site to show all 10 sites running at once, but part of it was that if we did have a camera and maybe a thermometer to get a digital thermometer to get the temperature in the lab, and this was running all the time, and we had timestamps that from the protocol we were able to put up to sync with that camera, then if something weird happens and we have to go back and debug, at least there's a video you can say, oh, in run 12 somebody bumped the machine and it no longer got that reagent or something. What other kinds of things other than a video would you want to have in kind of this this validation step, do you think?
So this is kind of a question to Rick, but I'm wondering if there's like logging at the level of that you can do at the level of PyLab Robot or OpenTron API, where you can log every command sent to the robot and match that with a video timestamp or something.
We don't do this right now, but this would be super easy to add. So yeah, I like the idea of tagging every operation in the video and then making it easy to scroll through that. YouTube has a similar feature where you can have like timestamps that are clickable. Great suggestion.
Some of the hardware, like I think PyLab Robot supports TCAN, and they have a message bus that you can tap into, which gives you really low level granular details about what happened. It's like internally it's a CAN bus. You just basically get all the details. You can get position of every gantry, you can get every measured dispense and everything. You can tap into that. You'll end up with gigantic log files, and then you won't know what to do with them. But there are, depending on, this is highly hardware dependent, but you can tap into these kind of log streams for some of the more commercial device. I don't know about the OpenTrons.
I think we should be able to get most of that information, everything that is available just through PyLab Robot. One hardware limitation is the OpenTrons doesn't actually know where the head is. That is why you need to home all the time, because there's no sensor that's going to tell you the head is at X, Y, and Z. This could be an issue, but I found that just by homing every time at the start of a protocol, and then every time you hear this OpenTrons make the horrible sound when it crashed into the wall, that is most reliable.
Hey, I just want to get us on a quick time check. We're at the top of the hour here. I think we should probably, I mean, I can stick around a little longer, and actually I don't want to interrupt the conversation, but I'm sure some people have to go. So I don't know if there's any sort of, we didn't get through half of this stuff, which is a good thing. But I don't know, any closing thoughts that you had in mind, Derek? And then we can kind of go into overtime if people are want to stick around.
No, really just to set the idea of this being a way of getting to critical mass in lab automation so we can extend it. I think if I were to dream forward 2 years, we'd have every one of these robot sites having an open enzyme production mechanism with them and a set of known reagents that were there, and anybody else in the country that site is in able to remotely submit jobs to it and get sent samples with some standardized MTA to return materials from that site within country, and that this could really seed this network of community lab automated biology sites that it can open this up to remote participation in a way that we don't have today. And we've got an awful lot of steps to get to that, most of which are not strictly lab automation steps, but the legal and infrastructure and associated equipment steps, everything that surrounds this. And so that's kind of where most of the last questions on there we're getting to. Is just this glorious future a few years down the road.
Yeah. I mean, just kind of give me a nod or whatever. I mean, are people interested in kind of continuing this conversation at some point down the road? I found it really interesting. Yeah, I see a thumbs up. All right. Well, let me know. I'll do it all off the number of emails I get saying I liked that. We should keep going. But yeah, this was great. Yeah, I have more thoughts, but I'm trying to actually wrap up. Anybody have anything else to add? All right, well then I will say thank you. I'm gonna stick around. If everyone has to go, that's all right, but I'll stop the recording and we can stick around a little longer, but otherwise, you're free to continue your days. Thank you, guys.